Welcome everyone to the FABI International seminar for this month. And our speaker is Malia Gehan from the Danforth Center. I met her online mostly, from talks, and I was invited by them to speak there, so we decided to reciprocate. Malia does some awesome plant phenotyping work and coding for plant phenotyping, and also has some nice projects on quinoa and flowers and heat stress and how it affects flowers. I must apologize, Bernard sends his apologies, he won't be able to make it because he's in a board meeting today. And is there anything else I need to say? Otherwise, I'm going to hand it over to Malia. You can share your screen and go for it.
Thanks, Nikki. Thank you for the invitation, and to everyone who helped organize my talk. So I'm going to talk today about what phenotypes matter, some open challenges we have in plant phenotyping, and I'll introduce the software that we developed called PlantCV. So I just want to point out, this is the institution I work at. I'm at the Donald Danforth Plant Science Center in St. Louis, Missouri. We're a nonprofit research institute, so we're not a university, we're not industry. I think we're the largest nonprofit plant science research institute in the world. So we have about 400 employees at the Danforth Center. So the main focus of my lab is, an interest of my lab biologically, is crop resilience to temperature stress. So we're interested in the environmental factors that might limit a crop's ability to grow. So both latitude and longitude, what are the main factors, environmental factors that influence or limit a plant's growth? We're interested in high temperature because of climate change. And we're also still interested in low temperature because it often limits a plant's seasonal growth. So if we were able to, for example, make a plant more cold tolerant or freezing tolerant, so a plant like crop like maize that's relatively wimpy when it comes to cold, that if we were able to grow it earlier in the growing season, it might be less likely to hit drought and heat later in the season, where it's more detrimental to the reproductive growth of that plant. So the way my group approaches questions of temperature stress really is to look at and utilize natural variation under stress. And so when we're looking at natural variation of stress responses, that often requires us to look at a large number of plants. And I know many of you are probably familiar with having to manually phenotype plants or phenotype plants in high throughput. And so if you've ever done any sort of manual measurements of plants, which I'm sure many of you have, it can be tedious. And so if you're doing large populations, especially if you want to associate a particular genotype with a phenotype, that can take a lot of effort. On top of that, we're also interested in the relationship between stress and time. And that's because if you give a plant, say, a stress treatment in the morning, you can get a very different transcriptional response than if you give it the same stress treatment in the evening. If you give a plant a stress treatment early in development, you can get a different response than if you were to give that plant the same stress treatment later in development. The duration of a stress matters. So an acute, very short stress can get a different response than a very long duration stress. And the time in which you phenotype a plant matters. So if you were to give a plant a stress treatment and look hours later, you might not see any response, but if you were to phenotype that plant days or weeks later, you might see an actual phenotype. So because of this tight relationship between stress and time and our interest in phenotyping a lot of plants, there's a lot of reasons why we were interested in non-destructive image-based high throughput phenotyping. So this video is showing, we have one of those fancy schmancy LambdaTech phenotyping facilities. Ours is called we call it the Bellwether phenotyping facility. So this can house 1,140 plants. It can water and weigh each of those pots multiple times a day if the person designing an experiment wishes to. And then the plants travel on a conveyor system out to imaging stations where they can get imaged, just RGB imaging. So just like your cell phone type imaging with a regular camera, or near infrared imaging, which can tell us something or estimate something about, say, the water content of that plant. So this system's very nice. We can get very high quality images of individual plants because these individual plants are moving to those imaging stations. But it's quite expensive even for internal users to use the system. And so my lab, when we first started, invested a lot of time and effort in installing very cheap cameras and computers into our growth chambers. So I don't know how many people are familiar, but we use Raspberry Pi computers. They're about $35 computers that you can have $25 camera modules hooked to. So quite inexpensive. And so with those camera systems, we've put them in growth chambers above flats. And so we can simultaneously image our plants through time. So this also has another advantage. So with the growth chambers, that means we can have multiple environments. So with the bellwether system, there's only one chamber. So you can only have one environment. And so if we wanted to do a temperature experiment, we'd actually have to do 2 experiments back to back. But with these Raspberry Pis, because they're inexpensive, we can set up the growth chambers with multiple environments. And we can also image lots of plants simultaneously, or relatively more plants simultaneously rather than individuals. We also have a hyperspectral phenotyping system. And with this imaging modality, we can get more information about plants. And I'll give more details about what hyperspectral imaging is a bit later in the talk. So we have all these fancy systems. And I was telling Nikki before this, our lab, we're sort of agnostic when it comes to the plant species we work on. So just in our lab, we have projects on Arabidopsis, brassica, brachypodium, maize, quinoa, rice, Atarian sorghum currently. And although our main interest is in temperature stress, so mainly cold and heat, sometimes we have projects that have combinations of stresses, so cold and heat in combination with drought or in combination with nutrient stress. And sort of this is just our lab alone. There's other labs at the Danforth Center that are working on large projects that are on different plant species or different stresses. And so with the variety of data that we're collecting on all these different species, we very quickly realized when we first started doing plant phenotyping that we needed the software that we use to analyze the images that we collect to be flexible and clearly defined so that we could easily publish them. So back in 2013, when we started, or I guess we're close to 10 years now, in 2013, when we started, there were some open source plant phenotyping software tools available. Often they were not editable, so they weren't open source or open development where we could change them. They were kind of black boxes. They were meant for specific plant architectures or plant only extracting a sort of single trait. So they were a little bit less flexible than what we were looking for. And then the commercial software that we had available to us was also not very flexible. And it was designed by people who very, very smart software engineers, but who didn't necessarily have the domain knowledge in plant science necessary to define the traits that were most important to the people who were doing the research. So just an example, the software engineers had defined this orange line as plant height. And they're not wrong because there's plant material up here necessarily, and there's plant material down here, but that's not how a plant biologist might define the height of a plant. A plant biologist might say, like, I have a boundary line here, and then anything above that line, let's call the height of the plant. And we might want to do, if we're doing a drought experiment, we might want to use that information as a proxy for drought. So how much plant height is above the line versus below the line, how much plant area is above the line versus below the line. And so that's really why we started to develop PlantCV almost 10 years ago. And so the vision of PlantCV really was to develop phenotyping tools that be modular and reusable so that they could be combined and recombined with ease, to build flexible workflows to quickly extract biologically relevant data from images and sensors. And these tools needed to be easy to use so that data scientists and biologists could use them. Not every lab has the luxury of having somebody with computational expertise, but every lab might have a project that requires some sort of phenotyping. And so we wanted these tools to be accessible to a wide variety of researchers. So at its core, PlantCV is a library of modular functions, and that's done very purposefully. We wanted you to be able to create workflows that fit your data. So if you have a very simple image, so by simple, I mean if you had a plant on a pure white background, that your image is perfect, then you could have a workflow that segments that plant out from the background that's very short because it's a very simple image. But if you had a more complicated image, say there's a lot of shadowing or the lighting is not perfect, or there's, somebody's hand got caught in the image or something, those images aren't perfect, then you might have a more complicated workflow, but you could still use the same tools. Within PlantCV, we then, once you have developed the workflow, made it so that you can then parallelize that workflow over a large set of images. And by large, we mean, you could have tens of images, thousands of images, hundreds of thousands of images to then use. And then what we extract, what you get out is a database that's of your extracted measurements, any associated metadata. So again, we don't want to reinvent the wheel. We don't develop PlantCV in a vacuum. Wherever possible, we are compatible with other open source imaging packages like OpenCV, and we use other packages wherever possible. And so if you're interested in learning more about the details of PlantCV, definitely this is sort of the website, and we show it sort of multiple. I'll probably show it at the end as well. So we do have core developers for PlantCV. We've been very lucky to get support from different funding agencies to support the development of PlantCV. And so our job as sort of the core developers really is to develop the infrastructure necessary so that external collaborators can contribute to PlantCV as well. So in order for PlantCV to continue on and be a sustainable software project, we don't want just people from the Danforth Center or even just our core developers developing functionality for PlantCV. We want the users to be able to drive what is important for the plant phenotyping community. And we want, so we want to hopefully build something that's sustainable that lots of people can use. So we have PlantCV is openly developed, meaning the latest tools and versions of PlantCV are always available. We do releases. That's usually done so that people know there's sort of a working version of, this is the release I'm using for publication. We're on GitHub. Most, if not all of our code requires testing. And what that means is that if I push code to PlantCV that breaks existing code, so if it breaks something, then I can't push that without fixing those conflicts. And that's very important. So because if we have external collaborators, that means that we can actually accept external code without it breaking everything for everyone else. Also really important to us and required for sort of adding tools to PlantCV is that we have very extensive documentation. Like I mentioned, we want these tools to be accessible to biologists and people with maybe less computational experience. And your software really is only as good as your documentation. So if people can't use your software, then it's useless. And they will only know how to use your software if it's well documented. And so we spend a lot of time on both static documentation and interactive documentation, and building tutorials to use the software. To actually download and use PlantCV, we're available on PyPy, Docker, and ContraForge. And we've, I think, really improved sort of the installation of PlantCV. It's become a lot easier. So again, this stuff isn't necessarily stuff that our users might see, but it's a lot of what our developers work on. And that's really so that we're building the infrastructure so that our external collaborators and users don't have to. So PlantCV, I'm not going to sort of go over all of what PlantCV can do, but in this talk, I want to highlight some of the things that I think are maybe going to be most useful to those interested in plant science in general. But we, when we first started PlantCV could analyze RGB images. So these are just, again, these are images that your cell phone might take, or a regular digital camera might take, grayscale images, and these near infrared images, because again, we had that fancy system, so we wanted to be able to analyze those near infrared images. We also can analyze fluorescence images, and then newly added is the ability to analyze thermal images as well as hyperspectral images. We have everything within PlantCV to be able to sort of segment plants using classical computer vision methods. There's some machine learning methods that have been implemented that I'll kind of highlight, and graph-based segmentation methods. So this was something that a lot of users had asked for, and that Haley helped to develop. So people wanted to be able to say segment leaves from stems and measure leaf angle. And so Haley added a morphology subpackage to PlantCV to make that possible. So we're often asked by the people who help to support PlantCV, like the funding agencies, how many people use it. And as an open source project, it could be difficult to say because you're not always familiar with who's downloading or using your software. But what we can say is that we have about 2,500 downloads per month on PyPy. This number is actually a little bit out of date. We have 36 contributors to PlantCV. We have 7 developer papers, that's still accurate, but now we're up to 52 user papers. And these are not papers just that are of people that are citing PlantCV, but these are people that are using PlantCV for their methods, for their research. So they've said, like, we've used PlantCV to do, XYZ in their project. So we're really proud of that number because that means that, people are effectively using PlantCV sort of in their research. On the left side here, I'm showing some of the interactive documentation that's been added to PlantCV within the past few years. So you can actually, without having to download PlantCV, use some of our tutorials, change parameters, and see what a workflow looks like. So we're incorporated with or compatible with Jupyter. So you can use Jupyter notebooks to actually get feedback on how is the segmentation of my image working? Am I very effectively extracting my plant and my plant traits that I'm interested in? And so that's been added in the past several years, and we think is important for people who are learning how to use PlantCV. So this is just some examples, not from our lab, and actually not often from Danforth Center even, of people who are using PlantCV for their research. There's a lot of people who are interested in machine learning or using deep learning or convolutional neural networks. And but they need training sets for those things. And so people are often using PlantCV as a pre-processing tool to develop those training sets. Or they're just using PlantCV for their research. We have some people who are looking at herbarium specimens, for example, or looking at corn kernels, or just images that they take in, looks like maybe in some sort of homemade imaging box. So these are sort of the examples that we might have expected people to use PlantCV for. They're using it for their plant research. But then we've also had some surprising use cases of PlantCV that we never expected. And so this paper came out in 2021, a different one came out in 2021. So this first one on top, they're actually using PlantCV to look at and measure cracks in sidewalks. And this one down here is looking at vesicle branching. So we've had some surprising use cases for PlantCV. These people never reached out to us, or these researchers never reached out to us, but they're clearly using sort of PlantCV in ways we never expected them to. For us, we find that PlantCV is a useful tool for education and outreach, especially when trying to teach younger researchers about data science and bioinformatics. And that's because we were traditionally teaching those tools and skills surrounding, say, sequencing data or genomics. But there you need a lot of domain knowledge, right? You need to teach a student, what is DNA, what is sequencing, why do you need to assemble a genome, sort of all of that sort of background knowledge before you sort of get into the sort of data science examples. With PlantCV, the application is a little bit maybe more direct, where we say, we're interested in extracting the plant from the background. So we don't want the color card or the ruler or this text down here. We just want to extract the plant and the plant traits that we're interested in. And so we find that PlantCV and some of the tools that we have developed are useful for education and for outreach to students. So like I mentioned, PlantCV is an open development project, and that means that when we first developed PlantCV, it was using data often from these commercial platforms that are expensive, but not necessarily accessible to many people. And so a lot of the tools that we're developing now or have developed in the past few years have been focused on enabling low-cost phenotyping. So again, phenotyping with these Raspberry Pis, and also enabling extraction of data from other imaging modalities like hyperspectral and thermal, just some examples. And so I'm going to go over some of the tools that we feel help to enable low-cost phenotyping. So this isn't necessarily a new tool. This came out, the naive Bayes classification tool is a machine learning tool that we use very often for plant segmentation or plant classification. So Arash, when he was in Noah's group, developed this tool where he wanted sort of binary, where he wanted two categories. He wanted either plant or not plant. And that's because you can, even in a perfect imaging situation, like in the lemon tech that produces images that look like this, you can still have fluctuations in plant color or lighting. And so you might want to have, instead of just using a threshold, use some sort of classification so that's a little bit more robust to that variation. And so this naive Bayes classification calculates the probability that a pixel belongs into one class versus another. And so in this case, we had two classes, plant or not plant. And so then we can use this naive Bayes classification tool for segmentation of this image. And by segmentation, I mean extraction of pixels that are just sort of associated with the plant. That's why this tool was originally developed. But again, we try and make PlantCV modular so that it's not we can use tools for multiple uses. And so we had a collaborator up at the University of Minnesota, Katie, who was interested in, she sent us these images of wheat, and they are experts in plant pathogens and plant disease. And then their experts were categorizing these scans into different sort of disease severities. So they were giving a score between 1 through 5. Basically, there's most severe being 5, least severe being 1. But Katie showed us these images and she asked, can we extract more information rather than just 1 through 5? And also, can we have that information be more quantitative? And so she wanted to classify or to quantify, say the number of rust pustules. They have this surrounding yellowing chlorosis around those pustules, as well as the amount of healthy tissue. And so again, with PlantCV, the training for this machine learning module is very fast. You can do it in about an hour or less, depending on how many categories you have. And so in this case, we had these 4 categories: plant, background, so like any plant pixel background, and then those pustule, and then the chlorosis. So plant by plant, we mean healthy plant tissue, so that green. So now with the output data, you can get the area of those 4 classes, and you can see it does a relatively good job getting even the surrounding chlorosis, which can be hard for even experts to sort of see. And so we can get a lot more information out of using this naive Bayes classifier rather than having even the expert score. It's also sort of a non-biased way of scoring the image data. This naive Bayes classification tool has also been used not just for plant disease, but for plant stress. So Tara Enders out of when she was in Nathan Springer's group, used this to quantify chlorosis or damaged tissue on maize plants exposed to low temperature. And she looked across sort of a population of maize. She also looked over time. So with one tool, we're looking at like 3 different applications of one sort of module. And again, it's nice because it's kind of robust to fluctuations in lighting, because again, it's calculating the probability that a pixel belongs to one class versus another, rather than a threshold that can be a little bit more restrictive. We've also have new thresholding methods that make it easier for users to threshold their images if they're interested in a little bit more traditional thresholding. This is a new thresholding method, a 2D segmentation method that Jorge has recently added to PlantCV. So when you're often doing have an image, you might want to threshold that so you just get the plant pixels. If you were to just so this plot is basically every pixel in this image plotted for their blue channel versus their green channel. So it's an RGB image, so we're taking the blue channel and the green channel. So if you were to do a threshold here on the green channel, you would get an image like this. And you see that you have isolated the plant in white, but that you also have a lot of background, so that mesh is sort of coming up as object. So it's not doing a very good job of just segmenting the plant. If you were to do a blue channel threshold and do a sort of cutoff that looks like this, where you're just trying to get this plant area, you get something that looks like this, where you still, you're getting the plant, but again, you are still getting a lot of background. And so with this new dual threshold method that has sort of interactive components, you can set a line that looks like this. And so you can very nicely isolate the plant in one step. So as a part of this, obviously it's easy for a user to visualize that with this graph and sort of pick where your thresholds are. And what was required for this was also to make interactive annotation of the data sort of necessary. And so that's something that has been a focus of PlantCV lately, has been adding these interactive data annotation tools. So within a Jupyter notebook, when you're building your workflow, you can actually sort of click on your images to say define regions of interest or to annotate data. And that's important because I think I mentioned several slides ago that people are obviously interested in using machine learning methods, but those machine learning methods, again, require you to have some sort of training data or labeled data. And that is often the bottleneck because people don't have labeled data. And so if we have tools that make labeling data easier, they might be more likely or able to use some of those methods, those machine learning methods. Again, speaking of machine learning, one of the biggest problems, again, is not having training data. So again, we wanted to develop tools that make it easier to either analyze data right away or to create training data for future machine learning. So Jorge developed this 3D watershed algorithm and has added it to PlantCV. So this should look sort of familiar. So this is plants in a flat sort of layout that have been imaged by Raspberry Pis from the top. So if the plants aren't touching, then PlantCV can do a good job of sort of segmenting those tools with the tools that we originally had available. But once plants start to touch, the computer's not smart enough to know that that they're different objects or different plants. And so previously, when we had an experiment, once the plant started to touch, we couldn't analyze data anymore, or be very difficult. You'd have to go and hand segment images. And so with this 3D watershed algorithm, it applies the watershed algorithm spatially and across time so that it can propagate labels from early images when they're not touching to later images when they are touching. So this is without really any training data. So it doesn't take time to for somebody to hand draw where those boundaries are. And so you can see in this last image here that it does a fairly good job. There's some pixels that are labeled here, say in this orange and green plant, but otherwise it does a fairly good job of segmenting the plants without any training. So this is great. So you could use the watershed segmentation to get, to sort of segment these plants, and then say if you're interested in machine learning, input that data and try and get even more information out of sort of later time points. Or just use this data as is and get just more data out of your data set. So this is using that tool in practice. So we had Soteria growing. So this is just to show that this tool doesn't just work on Arabidopsis. It's kind of agnostic to plant species, which PlantCV generally tries to do. We're not developing tools just for a single plant type or plant architecture. So this is great in our opinion, but another challenge is that once you segmented those plants out, you might want to try and get even more information than just the plants as a rosette in the case of Arabidopsis. And so how can we get more information out of this very inexpensively acquired data? So this second panel is using a convolutional neural network to do leaf segmentation, and this does require training data. But a lot of labs have shown that you can use this Mascar-CNN approach to segment leaves, and this again requires training data to use this convolutional neural network. So you can see here that it does a pretty good job of segmenting these individual Arabidopsis leaves. But one thing you'll notice is that the color is kind of jumping around. And that's because leaf 1 in image 1 is not necessarily the same as leaf 1 in image 2. So they're not linked through time in any way. And so often people have stopped here. They say like, we've segmented the plants. And then somebody has to manually go in and say like, okay, this is leaf 1, this is leaf 2. And so we wanted a way to sort of automatically assign leaves through time. And so this is this time linking module that's been added to PlantCV. And so now you'll see that these colors aren't jumping around, and that's because those segmented leaves have now been linked through time. And Danny has shown that this works not just on this data set, but on a data set for bean. And so we think we're trying to test it on other species with training data so that we can show that it's robust to species and architecture. So this is great because that means you get information about individual leaf growth, leaf emergence. And so we're getting more information out of this image data than just sort of the rosette as a whole. And so what's nice again is that even though we've already collected this data set, we can go back and we can extract more information out of it at a later time. That's sort of one of the powers of non-destructive and image-based plant phenotyping. Okay, hopefully that, yeah, there you go. So in addition to getting more information out of just those RGB images, like I showed in the last slide, we're also getting more information from images by using different imaging modalities. So again, RGB images, those are what you guys will probably have in your pockets with your cell phones. So that's a red channel, a green channel, and blue channel, so 3 channels. And those cover a wide range of the red spectrum, the blue spectrum, and the green spectrum. With hyperspectral imaging, instead of wide range of wavelengths in red, green, and blue, you can get discrete wavelengths depending on how good your camera is. So our camera has 1,000 channels within between 400 and 1,000 nanometers. So we're getting sub-nanometer resolution for our image data. And you can get basically more information about the plant or whatever you're imaging, spectral information about your object in comparison to, say, just an RGB image. So again, instead of 3 channels like RGB, in our case, we have 1,000 channels. So people have developed and calculated different indices. That means that they think are associated with things biologically. So things like NDVI or NDRE, which is a similar index, can tell you something about, say, the health of that plant. And then this other panel, this WI, is a water index. I like to show this one because it might be small on your screen, but there's a higher concentration, according to this index, there's a higher concentration of water along the veins of these leaves. So you see a higher blue value there. So we can get more information sort of out of this imaging modality that might be biologically relevant. And so we've added the ability to analyze this data and extract this information using PlantCV. Now, sometimes you want to actually develop an index or develop something that might be specifically biologically meaning to your own project. And again, we don't want to develop PlantCV in a vacuum. We don't want to reinvent the wheel or recreate tools that exist out in the community, especially if they're open tools. And so as part of a new grant that we have, PlantCV is making sure that it's interoperable with some of these other open source packages, open source tools. So we're collaborating with Alina Zar's lab, the Machine Learning and Sensing Lab at the University of Florida, because they have a nice suite of tools, hyperspectral tools that are sort of available. And so now PlantCV is compatible with these hyperspectral tools. And so we can use the segmentation tools in PlantCV, as well as the analysis tools in Alina's packages. And so I'm going to talk about 2 vignettes before the end of this talk. This is a project we had on hyperspectral imaging, early disease detection, and we had using these new PlantCV and GatorSense tools that are now interoperable. So the question here was, could we identify in these hyperspectral images disease earlier than experts? And could we differentiate diseases with similar visible phenotypes? So here we are working in sorghum, and we had two bacterial pathogens, Burkholderia and Xanthomonas. We also infiltrated with a mock as a control. And on certain genotypes, Burkholderia and Xanthomonas bacterial diseases will create or will develop into a red spot phenotype that when you show to an expert can't differentiate it. They look like red spots. And so if you had both of those pathogens, happen to have both of those pathogens within the field, you wouldn't be able to say differentiate them. So again, in this experiment, we're infiltrating with mock or the pathogen, and then we're imaging those plants every single day until we visibly see until we see visible symptoms of infection. So when we're doing these experiments, we image till day 6 or 7. But you can start to see, if you really look closely at the plants, those visible phenotypes at day 4. So just keep that in mind. So again, here is the red spot phenotypes at day 6. Again, if you showed this to somebody who's extensively studies these bacterial pathogens, they can't tell the difference. So again, we use PlantCV to extract the spectra, and then we use the tools in the machine learning and sensing labs package, the GatorSense tools, to train a spectral match filter disease discrimination for spectral match filter algorithm, so that we could see if we could do early detection and disease discrimination. So we used the last day of imaging, so 7 DPI, to detect Burkholderia. So just keep that in mind. So this is what we use for training data. And so we're just showing here what the detection looks like on the training data. So this isn't super impressive because this is the same data set used for training and for detection. So this is day 7, and you can see what's nice about this is that we get detection, so highlighted in orange, of Burkholderia, but no non-specific detection of the disease in Xanthomonas. So even though an expert can't tell them apart, we can with hyperspectral imaging and the use of this SFMF detector. And we don't get any non-specific detection on mock either. Okay, but again, this is the same data used for training. So let's look at data that wasn't used for training and earlier days. So 5 days post-infection, we get detection of Burk on Burk, which is great. We get a little bit of non-specific detection on Xanthomonas, but not too much, and nothing on mock. And then remember, like I mentioned, 4 days post-infection is when an expert can kind of see Burkholderia, so we wanted to go earlier than that. And so this, again, here, what's nice is we get detection of Burkholderia on Burk, a little bit of non-specific, again, but not much on Xanthomonas, and nothing on mock. So what this suggests to us, even though this is quite preliminary, is that early disease detection is possible because it's earlier than what experts can tell at day 4, and disease discrimination between Burkholderia and Xanthomonas seems possible. So this was a very exciting result for us. And we're sort of following up. So if there's anybody out in the audience who develops or develops open source packages for plant phenotyping, it doesn't need to necessarily be image based. Please feel free to reach out to us, because we have this new grant funded by the USDA to help to make PlantCV and these other software tools important for the plant phenotyping community interoperable. So again, I mentioned and highlighted the interaction between PlantCV and the HSI toolkit from Alina's group. But part of this grant is to make this project interoperable with other ones, as well as add new functionality. Okay, in my last few minutes, I'm going to talk about a use case of PlantCV in our group. And that is to look at quinoa under heat stress. And I like to use this project as an example because often when you start a new project in plant phenotyping or plant phenotyping under stress, you don't know what phenotypes are going to be the most important for the plant. So what traits are going to be the most affected under these stress conditions. And so this is the case of our work in quinoa. We started this work when I first started my lab in 2016. And we had postdocs that were interested in looking at differential heat stress between the roots and the shoots of quinoa. So quinoa we are interested in because it is a plant that can grow in quite diverse environments. You can water it with salt water and it thrives, or half salt water and it thrives. It grows on salt plains. And there are varieties that grow at very high altitudes, so really high light, but very low temperature, so quite freezing tolerant. There are subtropical varieties. And so quite diverse environmental conditions that different accessions of quinoa will grow in. One of the main limitations to expanded growth of quinoa is its heat tolerance. Even though there are subtropical varieties, growing quinoa in the US is mainly limited by its heat tolerance. And so I had postdocs in the group that were really interested in looking at quinoa under control conditions, so same conditions for the roots and the shoots, heated roots only, specifically because quinoa is supposed to have a grain that's very nutritionally rich, and so we were interested to see if specifically heating the roots had any impact on the nutritional quality of the seed that it's produced. Heated shoots only, or heated roots and shoots. And how they did heated shoots only was they ran a cooling coil in the soil, and how they did heated roots only is they had a heating coil for the soil, so quite technically challenging, and they did a lot of work to make sure the setup was right. So they started the heat treatment at anthesis, so at flowering, and they did that heat treatment for 11 days and collected phenotype information during that stress treatment. So again, this we're doing at a much smaller scale than you would in one of those in commercial platforms. And again, we didn't know exactly what would happen under these stress conditions. We hadn't done any. This is what we weren't sure what's going to happen. So what we found was that total yield is reduced in any treatment with shoot heating. So the heated roots and shoots, or the heated shoots only, had a significant reduction in yield. And the heated roots only had no reduction in yield in comparison to control. And this wasn't necessarily expected, because if you do the same sort of treatment in wheat, you get a different result. If you heated roots specifically, you get a significant reduction in yield. And then, so we weren't exactly sure what quinoa would look like. The yield for quinoa is made up of seeds from the main panicle, secondary panicles, and tertiary panicles. And so if we just look at yield from the main panicles, it looks very similar to like the total yield. So most of the yield for quinoa is coming from that main panicle. When we were looking at those panicles and phenotyping those panicles, we noticed that, again, in those samples that we had reduced yield, they looked less mature than the samples under control or heated root only conditions. And so we wanted to explore that a little bit more. So again, we used that naive Bayes classifier that I mentioned several times throughout this talk to quantify the maturity of those plants and the biomass of those plants with heated shoots and also heated roots. And so what we saw again was that anything with heated shoots had less yellowing plant area. So those plants were likely less mature, and they also had more biomass in comparison to the control or heated root samples. So those were further along in their maturity at the same sort of days after planting. So even though those plants were bigger that had the heated shoots, even those plants were bigger, they had less yield from their main panicles and also from their secondary panicles. But again, what was interesting is that we saw that there was actually an increase in both the number of tertiary panicles with the heated shoot treatments, as well as the yield of those tertiary panicles. So that's interesting. So there's a change in resource allocation in any of the plants with heated shoots in comparison to even the control or heated root samples. So unfortunately, farmers don't wait until the tertiary panicles are mature for them to harvest. So this is not agronomically practical to increase yield under heat stress. So we still have to figure out a way to increase the yield of those plants under heat. And so we were doing transcription. We did an RNA-seq experiment on plants under those heat stress conditions. And we found that there are about 400 genes that were commonly differentially expressed in the samples with reduced yield. Out of that 400 genes, there were 10 transcription factors. All of those transcription factors had something to do, I think all but one actually, had something to do with flowering. And one actually, RF2, you see a reduction in yield in plants like Arabidopsis because of a change in flower opening and closing timing. So this result from the RNA-seq led us to look at flowering more closely. And what we saw, and what Jose saw, because he really did all this work, was that there was no change in the numbers of flowers, and it's quite tedious work to count quinoa flowers. But there was a change in the number of flowers forming fruit. So not a terribly surprising result, but it was interesting that there was no change in the development number of flowers that were developing, but the number of flowers forming fruit. And so what he then looked at was changes in timing of flower opening. And so flowers were open for a lot longer under these control or heated root conditions where there are no changes in yield in comparison to the heated root and heated shoot conditions. And so under control conditions, you can see those flowers are open and the anthers are spread, but under the heated shoot conditions and the heated root and shoot conditions, those flowers are closed. And so those anthers are sort of tucked into the flower. So you can see that better on this bottom here, open versus closed. And so our hypothesis is that as a mechanism to protect those flowers from heat, they stay closed longer, but they're less likely to get pollinated and form fruit. So we're trying to use now hyperspectral imaging to screen a larger population for this phenotype because counting flowers is quite tedious, and counting open versus closed flowers is even more tedious. And so our idea is that you can nicely pick out the anthers from hyperspectral images. So can we more easily see spread open anthers in hyperspectral images versus closed ones? So that's how we're going to screen, hopefully screen a larger population for this. Our lab has also developed a pollen germination protocol. So, the pollen, in addition to not being able to be accepted into the flower because it's closed, the pollen itself might be less viable. And so we've developed the pollen germination method that was recently published in Plant Methods. And we've also developed PlantCV tools to enable the annotation of germinating versus non-germinating pollens that we can look under heat conditions. Just as an last two slides, I mentioned at the beginning of this that we weren't sure if there were going to be changes in the nutritional quality of the seeds collected under heat stress. So even though there's no reduction in yield in the heated root samples, we do see differences in the inomic profiles of those seeds, even though the yield is the same. So sodium, like I mentioned, is an interesting one under heat stress because, again, quinoa is known to be salt tolerant. What we find is that the impact of sodium, there's a decrease in sodium concentration under heat stress in a comparison to control in all of the samples, even though these heated root samples didn't see any decrease in yield, suggesting that there could be an interaction between heat stress and maybe salt tolerance of these plants. We don't know that yet. And so this is a future direction going forward that Lucia is going to be pursuing as part of a USDA fellowship project. So just in summary, we developed a tool called PlantCV that we hope lowers the cost barrier for high throughput phenotyping. And we're developing tools to try and get more information out of the data that we're already collecting. Yes. So I just want to thank the people who actually do the work and the funding agencies, and to mention that we are hiring postdoctoral researchers. So if you have any desire to come work at the Danforth Center and work on plant phenotyping or plant stress, please let me know.
Thank you. That was great. There was some applause in the room. I didn't turn on the room. Thank you very much. In the meantime, I can ask a question so long. So I saw that you're planning on extending PlantCV, and there was a picture of a drone. So what work has been done on linking up to drone phenotyping?
Yeah. So there's a package called Field Image R that Philippe he's now at I think he's at Syngenta or Corteva. So because he's moved on to a company, he's kind of passing the torch, and he's actually passed the torch to our team. And so now we're helping with maintaining this Field Image R package, which is a great set of tools. And our plan that we've talked to him about is really to make those tools interoperable. So, and some of the functionality is already available in PlantCV, and we actually have a student this summer, a very talented REU student, who's working on using PlantCV to analyze that data, and then where it's not possible, create new tools to do that. So a lot of it is in, say, making sure that we are able to segment the plot level data effectively. And then we're also providing maintenance support for that Field Image R package. Yeah, so mainly we haven't had that data in the past, and so that's why it hasn't been the focus. But now, I think I mentioned to Nikki specifically that we're having more field work and more fields, and so it's becoming more of a shift in focus towards that type of data.
Are there any tools for thermal imaging with the drone data or not yet?
We have tools for thermal imaging. It's been in chambers, but again, you could use the thermal tools. There's no reason why you couldn't use the thermal tools on field data. Yeah, so some of the tools specifically added for thermal have included ones for like image registration, which is important when you're often trying to do segmentation off a grayscale image. And so there's no reason why you couldn't use those tools in addition to the sort of segmentation tools.
Okay. Are there any other questions? My team here, do you guys have any questions? You send your fiancée to go and visit. She's doing some drone imaging with multispectral, not hyperspectral.
Yeah, we there's a new geospatial institute that just opened St. Louis that Danforth Center is a part of, and so they're collecting a lot more of this type of data, and so we expect to be developing more tools in that sort of area in the near future.
And if no one else has any questions, I'm just going to keep asking questions. So, do you have any questions? No. So it's just nice to see some of the disease imaging stuff and that's moving in that direction. It looks quite well thought out. Like I like the being able to look at the different diseases with the hyperspectral analysis. Is that tool applicable across many different diseases, or is it not a tool yet?
I think it would be applicable across many diseases. I think you would need to do training and have training sets, obviously, for whatever disease you're looking at, which can be tedious to collect, which is the case for, again, the case for anything that's going to require training data or using these algorithms.
What kind
but the methods are, I think, applicable. Sorry.
What kind of size training data sets do you need?
What we've found so far is that if the phenotype is quite obvious, we don't need a lot of training data. But if it's a more subtle phenotype with less variation, then we usually require more.
And is there like a limit? Like 50,000 photos?
So at least with the hyperspectral, the data sets are quite small there. Like we're talking about tens of images, but they're quite data dense within that 10. So like I think each of those images is 50 gigs. So it's collecting quite deep data, but not a lot of it. If you're doing something like morphological, I could see that requiring a lot more training data. So like the MASCAR CNN, like those data sets are much larger for training, like hundreds of images or thousands of images that have been segmented. And so those are quite a lot larger, but the actual data is quite small. Individual images.
Okay. Yeah. So I had another question about field using. If I would take my camera into the field and take pictures, is the background a big problem for these tools? Because a lot of the things you showed were in these nice little chambers.
So that's a great question. So we've seen, actually somebody tweeted, I didn't show the tweet, but somebody had something where they're using PlantCV and they just took an image out in the field, and there was space between their plants, and so they could just use the segmentation tools without anything. If you're interested in plot level data, I don't think there's necessarily a problem in doing the segmentation with PlantCV tools that we kind of already have built in. So it just kind of depends on the resolution that you're interested in. If you want to segment individual plants, then having different imaging modalities like LiDAR or something where you have 3D information is obviously very helpful. But yeah, I mean, segmentation is going to be a problem if the plants are overlapping, or you just have a lot of undergrowth or something like that, because you're gonna, green is plant or something like that. It kind of depends.
Oh, it's in the chat. It says, thanks for a nice presentation and the insights in the new capabilities of PlantCV. Could you go into detail how time series dependent approaches, 3D watershed/unique ID, fit with parallelization?
That's a great question. I personally have not used the watershed algorithm, but I know Jorge has used it. I mean, he's using it right now across thousands of images. So he's obviously using those parallelization tools. But I think he does need to have workflows for those sort of individual flats. So what they're doing now is making these PlantCV templates to make that a bit easier. So because what you do need is to have the centers, I think, of those. And so actually what David's doing is this summer, Jorge is his mentor. What they're doing this summer is to make those template workflows to make that much easier, so that it's easier once you have those centers, you just parallelize the data, and that's not a problem. But getting those centers can be a problem because, you know, with each camera, you're kind of getting shifts. So I think that answers your question. Once you have the centers, it just works. But getting those centers, they're trying to make easier.
Okay, thank you. Otherwise, thank you very much for an awesome talk. I might be hitting you up with some extra, especially the field image R stuff. And thank you for your time and for a lovely presentation. And I hope you have a good rest of your day.
Thanks. Thank you, everyone.
Bye.